daylight saving time. I hope you did okay with the 1 hour jet lag. Right, we're going to look at photosynthesis. Hey, I have something to tell you. So we'll finish the topic of photosynthesis today. That's certainly happen, and we can make a start with the actin cytoskeleton. But it would really make sense to finish the topic of muscle contraction, which is part of the actin cytoskeleton. And I don't think we can do that today. We can, but it can be really fast. And that's not good. So I think we'll have plenty of time to finish that on the Wednesday and then still review a lot. And what you have my promise that anything that comes by on Wednesday that you will need to know for the exam, you'll get an extra clear warning from me. How's that? Shall we do that? I know it's a tough week for you. Say it for me, by the way. So Kelvin cycles, what we're looking at. This is where the ATP and NADPH from the light reactions is consumed. It involves this important enzyme, rubisco. It's certainly not the only enzyme of the Kelvin cycle, but it is the major one. And the most abundant enzyme on the planet, for sure. And why is it abundant? Because it's not working very well, so we need a lot. That's the message. Now here are the reactions of the Kelvin cycle that you should not have to learn. They're interesting. And I'll tell you a couple of things. Here's an overview. Here's the main enzyme rubisco, ribulose 1,5-bisphosphate carboxylase oxygenase. It is named after the substrate ribulose 1,5-bisphosphate. That's the two phosphates. The phosphates serve to activate this molecule. There's an interesting resonance here. I can hardly see it on my screen. Maybe you can on this screen. And that resonance is used here for the attack here by this CO2, which is catalyzed by the enzyme. And then we get this C6 unstable intermediate that immediately splits in the 3 carbon molecules, 2 of them. So we call this the C3 type of photosynthesis, because carbon, if it's given radioactively, is first found in a 3 carbon molecule. And essentially, this is the only type of photosynthesis that really occurs. There are modifications of this, but this is the core. So this first molecule that is formed, the PGA, is further activated and then ultimately it is reduced, and then we get the end product of the Kelvin cycle, which is glyceraldehyde phosphate. That's glycerol, where there is also an aldehyde, so one of the OHs is replaced by double bonded O, and then it's phosphorylated. And this is what we call a triose sugar, meaning a sugar based on 3 carbon molecules. And this triose sugar, GAP, or glyceraldehyde phosphate, that triose sugar, here it is the end product of the Kelvin cycle. Now, why is the Kelvin cycle a cycle? Because the glyceraldehyde phosphate that is produced, some of that can be taken out and exported, put for other purposes to make carbon skeletons, but the majority of that triose phosphate has to be used to remake the starting point of the Kelvin cycle, the ribulose bisphosphate. And that takes a whole set of reactions here, which is in this step 5, which really is not one step, but these are multiple steps. This part of the Kelvin cycle here is certainly not unique to the Kelvin cycle. Effectively, we have those enzymes as well. We can do those reactions. These are the same reactions that are used to convert C5 sugars into C6 sugars and the other way around. So if we need to go from sugars that look like glucose to sugars that look like ribose, which we have in our nucleic acids, we have a way to interconvert those. We can shuffle the carbon around. And those pathways, it's called the pentose phosphate pathway, is essentially here. This represents a couple of enzymes. So overall, what you should take from the Calvin cycle is it is dependent heavily on this Rubisco enzyme. There's a carbon fixation phase. Then there's a phase where there's input of energy in the form of a lot of ATP and NADPH. That's where the reduction takes place. GAP glyceraldehyde phosphate is the end product. And then there's a carbon shuffle phase finally. So 3 phases. And importantly, you should know that for every C3 molecule that is formed, you of course need 3 CO2s. So for every triose phosphate, we need 3 carbon dioxide, that makes sense. We need 9 ATP in total, and we need 6 NADPH. This is something I think you should be able to remember. And what these numbers tell you is there's a pretty high energy input in this process. And of course, these coenzymes were charged in the light reactions. All right. So what happens with the GAP, the glyceraldehyde-free phosphate, that end product of the Calvin cycle? What happens with it is it can be exported, and if it's exported, it can be in the cytoplasm, converted first to glucose and then to sucrose in a set of steps. And sucrose, which is a sugar composed of fructose and glucose, is the main sugar that is transported in plants. So if you have, in a couple of months, if you tap your maple trees for their syrup, then the main sugar that you find in the phloem sap of those trees, the main sugar will be sucrose. Most plants transport sucrose, there are exceptions. So that's for export, and that sucrose goes to other tissues in the plant, and that's the energy and carbon source for those other tissues, the roots, new leaves, flowers to grow. Another possible fate is that if photosynthesis runs really fast in the light, then the triose can be used to make glucose, which is then activated and used to form starch. Starch is the equivalent of glycogen that we have in our muscles and in our cells. It's a polymer of glucose. So starch can be stored in the chloroplast itself. That starch pool builds up in the day, and then actually in the night it's broken down, and the resulting hexose sugars are removed from that cell and they're exported to other parts of the plant. Okay, so 2 major possible fates of triose sugar. One possibility stays in the chloroplast, becomes starch. The other is it's exported and it becomes sucrose. Good. Those were the light reactions and the dark reactions essentially. Now there's a couple of problems that we have to understand. In the process of respiration, you should remember that the whole process is driven by the demand for ATP. If you do work, respiration picks up. Photosynthesis is driven by light, and it produces sugars that the plant can need for various purposes. But it's very difficult for the plant to directly couple the availability of light to the need for sugar production. And this is because light is not regulated by the plant. There are clouds, there's variations in sunlight, there might be other leaves overhanging the plant. Or if you're an algae and you're in the sea, the way the waves break makes that some points in time you're in high light and other points you're in lower light. Photosynthesizers do not control the light that they receive. And so they need to make sure that they have some ways to be sure that photosynthesis runs well when there is a lot of light and at a lower pace when there is less light. And there are a couple of mechanisms for that. One is that light directly regulates the Calvin cycle. And you do not have to remember the mechanism, it's on this diagram, and I'm going to explain it very briefly, but the point is, light regulates the activity of the Calvin cycle enzymes. The mechanism is fairly simple. Photosystem 2 can not only reduce NADPH, NADP+, it can also reduce actually a protein that has cysteines, and those cysteines can be reduced. So we can have disulfide bonds or we can have reduced cysteine on there. And the presence of that reduced cysteine serves as a signal to directly activate another protein, which then activates 3 of the Calvin cycle enzymes. So essentially, in the light, the Calvin cycle is turned on. In the dark, all of these reactions are reversed, and the Calvin cycle enzymes become inactivated. So that's one thing. A second thing that plants can do is if there is a lot of light on a leaf, then the relative activity of photosystem 1 and 2 can be changed. When there's a lot of light, the light harvesting complexes from photosystem 2 are released and they're allowed to go to photosystem 1. And that gives relatively more cyclic electron flow, and that's a condition that is beneficial under high light intensity. The one reason why that is beneficial is actually point 3 here. Cyclic electron flow gives a very steep pH gradient over the thylakoid membrane, and that leads to actually an inhibition of the activity of photosystem 2. So when there's a very steep proton gradient, it's difficult for photosystem 2 to push electrons into the electron transport chain because plastoquinone is already reduced and it's hard to get it oxidized. So you have to pump against a very steep proton gradient, and that's hard. So under that condition, when the pH in the lumen is low, photosystem 2 has a mechanism by which you can lose light excitation energy in the form of heat. And that's a safety mechanism. And we need that safety mechanism because photosystem 2, because of its architecture, it has close together a strong oxidizer and a strong reducer, and that's a dangerous thing, because there's a lot of explosive energy there, if you want. Photosystem 2 can actually be damaged when we give it too much light. Luckily, all the photosynthesizers have evolved a mechanism to deal with this, because the damage is mainly restricted to one protein part, and that protein, the D1 protein, is easily replaced. But if that happens, it's still kind of wasteful. And then there's finally one mechanism that we know as photorespiration. I've been baking pancakes last Friday. There's one person in the room who knows that this is true. And when you're baking pancakes, you have a stove and heat on the stove, and you have your pans on there. And you want to make sure that your pans are not bursting, burning up completely. So what if you run out of dough, or something like that? Or actually, if you have a stove, this should be my analogy. If you have a stove where you cannot control the heat, and this is analogous to your plant and you're in the sunlight and you cannot turn the sun off. Well, even if you have not enough people to eat the pancakes, it's best to just keep going with your pans and throw the batter in all the time and just keep baking, even if you don't need all of those pancakes, Just keep going, because if you have too much heat in the system and you don't quench it with your batter, your cans will go bust. So that's hard. And that's kind of the problem of photosynthesis. I'm not sure if the analogy works. You might as well waste a little bit, but don't blow the whole machinery up. This is the message. Now, there's one issue in photosynthesis that we call the point of photorespiration. And I view this as a potential safety mechanism, but at the same time, it's kind of wasteful, and it's also an issue in agriculture. And it is due to the fact that rubisco can not only react with CO2, but it can also react with oxygen. And that reaction is depicted here in this diagram. So this so-called oxygenase activity, indicated with the red arrows, leads not to the formation of triose sugars, but it leads to the formation of a C3 and then a wasteful C2 molecule. So our 5-carbon ribulose 1,5-bis phosphate splits into a C3 and a C2. And the problem is that this C2 intermediate is in addition toxic. It's not something that you want to have around. So this byproduct, which happens if there's a lot of oxygen at the site of rubisco, needs to be removed, and it needs to be regenerated to a C3, which can cycle back in the Calvin cycle and become the starting point again. So this is just something that happens. The question is, why does it happen? Well, probably the reason is that when photosynthesis evolved originally, rubisco was a very good idea. Because CO2 levels were probably very high 3.5 billion years ago, and there was no oxygen, because photosynthesis hadn't produced it yet. In the next 2 billion or so years, oxygen levels still didn't rise, because iron would have bound all the oxygen. So CO2 levels were always favorable for rubisco, and during all of that period there was no selection on the enzyme to not have the oxygenase activity. And in those 2.5 billion years, the whole mechanism of C3 photosynthesis evolutionarily got fixed. And nowadays, rubisco just can't help itself anymore. The last billion or so years that the oxygen levels have been going up, it just had to deal with this mechanism here. So photorespiration involves is actually the regeneration of this C2 molecule into C3. And it is a long and interesting pathway, and you do not have to learn it. But I'd like to say a couple of things. First, this is a beautiful EM picture. It's a thin section, and it highlights a couple of points that we always tend to ignore in cell biology. So first of all, we see here a chloroplast, and note that it is huge. It's only part of the chloroplast. There's a peroxisome, and there's a mitochondrial. Look at the size of these organelles relative to each other. Already that's interesting. Look at how close they are together, and then look at this space here, which is actually the vacuole of that plant. We tend to, in our diagrams of eukaryotic cells, ignore the fact that organelles can be close together, can be in contact, and that actually the cytoplasm can, in some cases, be stuffed with them. Now, why are the free organelles in this picture? Because someone decided this is a good way to explain the pathway of photorespiration, and it involves the peroxisome. Why? Because the breakdown of glycolate from P-glycolate, that's the first intermediate of the oxygenase reaction, that glycolate has to be converted in the peroxisome because there's a reaction that produces hydrogen peroxide. And the peroxisome is equipped with catalase, that's this enzyme that you see here in this sort of matrix-like structure. The glycolate is then converted to an amino acid, glycine, And that just drives this reaction forward, and then in the mitochondrion, the two glycine are used to form serine. And then that's deaminated, and we get back to the starting point here of something that we can use in the Calvin cycle. So what you need to know is 3 organelles are required for the completion of photorespiration. That's the chloroplast, the peroxidome, and the mitochondrion. There is an extra use of ATP right here to get the phosphate on. And there's a loss of carbon dioxide. So overall there's a net loss. Still, in the summer, in many places where there is C3 photosynthesis, plants can waste up to 50% of their fixed carbon in photorespiration. So it's going to be a significant loss. So you can ask the question, is there any function? And I say, yeah, maybe there actually is, because it's better not to blow up the whole photosynthetic machinery. It's better to waste a little bit. So yeah, maybe that is a function. But we can also look at this as just an unfortunate consequence of the way in which photosynthesis evolved. Because it evolved without oxygen. And it got fixed a long time ago in evolutionary time. So, to get back to the point of the protection, if we make a plot where we have the light intensity out in the field and the photosynthetic output, so we can say that's carbon fixation, that would be my blue line. But we know at the same time, if we measure light absorption as a function of intensity, it's more or less a straight line. This blue line, you know what it represents. That's essentially Michaelis Menten plot. It's just an enzymatic system that is saturated. So you can look at the plant as a huge enzyme that takes light and photosynthesizes. And that's saturation. So obviously there's a lot of light that the plant can absorb, but it has to get rid of it. And this is one of the problems that photosynthesizers have to deal with. And photorespiration can be part of this solution, but there are other mechanisms, especially dissipation of light energy as heat in photosystem 2, which make that this process here happens safely, the dissipation of excess light. Because there is no way a plant can couple the light that it needs to the demand for photosynthesis. To give you an idea, probably in this room, I'm guessing that we are at a light intensity of some 50 to 100 photons per square meter per second. Micromole photons per square meter per second. That's also known as the micro-Einstein. So we're roughly at maybe 100 micro-Einsteins here. On a bright day in Colorado, out in the field, you would be at 2,000 to 3,000 micro-Einsteins. So 30 times more light than we have here. The saturation point for photosynthesis is at roughly 300 to 400 micro-Einsteins. Okay, so take a light intensity that's maybe 3, 4 times, 5 times higher than what we're in here. Photosynthesis saturates. But bright sunlight outside can be 5 times higher than that. Just to give you an idea. So photorespiration is a problem in agriculture. And it's a problem for some plants, especially if they're growing at high temperatures, and that means that there actually is a problem for them. Because at high temperatures, plants transpire a lot of water, and plants have little holes in their leaves that allow them to transpire that water. They have to close those stomates, as they're called, so that they preserve water. But the preservation of water means that they cannot let CO2 in. So then their photosystem 2 is still producing oxygen, and we're not getting good CO2 in, so then the CO2 to oxygen ratio becomes very unfavorable. So if it's dry, or if it's hot, And as a plant, then you need to conserve water. Photosynthesis becomes more problematic. So what is then a possible solution? There is one. And that's called C4 metabolism or CAM metabolism, which are two forms of photosynthesis where there's an addition to the C3 mechanism. CAM stands for crassulacean acid metabolism, and this is something that we see in, for instance, the cacti. If you've ever gone into the desert in the morning and you've taken a knife and chopped a cactus and you taste that leaf and it tastes acidic, what is going on there? What's going on is there's an enzyme called PEP carboxylase, and it uses a C3 molecule, phosphatidylpyruvate, pyruvate, and it couples carbon dioxide to it. So the PEP carboxylase, PEPC, catalyzes the coupling of CO2 to phosphatidylpyruvate, pyruvate, which leads to the formation of oxaloacetate. And oxaloacetate is a 4-carbon molecule, and therefore we call it C4 metabolism. And this molecule can be further reduced to form malate, which is what is accumulated in the cactuses in the night in CAM plants. The big trick is that the PEP carboxylase is not sensitive to extra oxygen. It doesn't have that oxygenase reaction at all. So it can just fix CO2 even at low concentrations. But there's no way in which this enzyme can be part of a cycle. So ultimately we depend on the Calvin cycle. CO2 has to be released and then is later fixed by the Rubisco enzyme. What then happens is at the site of Rubisco there's now a much better ratio of CO2 over oxygen. So these are CO2 concentrating mechanisms. What is the difference between C4 and CAM? In C4 plants, it happens in plants, the site where PEP carboxylase is separate from the site where the Rubisco enzyme is active, as seen in a moment. So the C4 molecule is transported from one set of cells in the leaf to another set of cells in the leaf, which generates in one set of cells the high concentration of CO2. In the CAM plants, in the cacti, what happens is a separation not in space but in time, where the initial fixation by PEP carboxylase is in the night, when it's cool and the stomates can be open, we don't lose water. And in the early morning, and in the day, that CO2 is released and it's fixed by Rubisco. But because of this complicated mechanism, a cactus always grows slowly. Because it can only deal with the CO2 fixed by PEP carboxylase in the night. Okay, so in both cases we have this CO2 concentration and it minimizes photorespiration. So in hotter climates, C4 plants tend to win out. And a lot of the plants that we see on the eastern plains in Colorado, a lot of the grass is there, for instance the blue grama grass, is C4. And a plant that does well in hot conditions, such as corn, is a C4 plant. And here is the overview of what happens in the C4 plants. There's a special anatomy. Around the vasculature, so around the veins, which is the site where sugars are later exported, there are special chloroplasts that have the Rubisco enzyme. And these cells here, called the bundle sheet cells, because they surround the bundle of vasculature, these cells have the Rubisco enzyme, but the cells further away, the regular mesophyll cells, don't have the Rubisco. They have the photosystem II activity, so they split water, form oxygen, and they form the oxaloacetate and the malate, which is transported to the bundle sheet. In addition, they reduce NADPH and that reductant is transferred into malate. That's a reductive step. So this more reduced molecule is transported to the bundle sheet. In the bundle sheet, that reductant is used to also drive the Calvin cycle. So the result is that in the bundle sheet, we have a favorable CO2 concentration, and we have a low photosystem 2 activity. The water splitting is also not happening in the bundle sheet. It's very low photosystem 2 activity here. So that's how the Calvin cycle becomes more efficient. Corn is an example. All right. I think that's photosynthesis for me. How are we doing, class? I know you have your lab reports due this week. Some of you have been approaching me with some questions about the lab reports. Honestly, today I am super swamped and busy, so it's not the best day to ask me a lot. I will have very brief time right after the class, but you really should ask lab-related things first to your TAs. That's all we have. All right. Now I have good news for you. And the good news is we're done with any topics about ion gradients and electrochemical gradients. But the other good news is that I think you have a good chance of understanding it, actually. All right. Okay, let's start to have a look at the cytoskeleton. What is the cytoskeleton? I'm a guy who likes membranes, and I'll give you my perspective. The cytoskeleton is what you see by accident. This is how it was discovered. I'll read you a story. It's what people were seeing by accident when they were trying to do EM thin sections, and they mistreated their cells. They actually used detergents, washed away the membranes. Hey, they're still seeing something. Cytoskeleton. This is a picture of the